So, good morning, everyone. I would first like to thank the organizers and Alpha for inviting me to give a talk. So, yes, I come from Rican Center for Developmental Biology, and I opened my lab there about a year ago. So, the work that I'm presenting today is work that I've done as a postdoc. So my lab is interested in understanding the fundamental mechanisms of how endothelial cells develop into an interconnected tubular network of blood vessels that you can see here. And the formation of blood vessels, and as well as the maintenance of blood vessels, is critical throughout the lifetime of vertebrate. During development, the formation of blood vessels support embryonic and tissue growth, such that inadequate blood vessel formation can lead to embryonic lethality. And in the adults, blood vessels support homeostasis by catering for the metabolic demands of tissues and organs by bringing oxygen and metabolites. And it also serves as a conduit through which immune cells are transported to sites of infection, and new blood vessels are also important in regenerative processes such as wound healing. There are several ways in which blood vessels can form, and one commonly used mechanism is called sprouting angiogenesis, which is a multicellular process that is tightly coordinated in time and space and requires many endothelial cell processes to shape the vascular network. So angiogenesis, by definition, is it's when new blood vessels are formed from pre-existing ones. And proangiogenic factors, such as vascular endothelial growth factor, or VEGF, then activate endothelial cells from the pre-existing vessel to stimulate tip cell formation. And tip cells are more migratory and invasive. They form many filopodia. And they spearhead the formation of new vascular sprouts. Endothelial cells that trail behind tip cells, they're called stalk cells, and together they migrate collectively into the hypoxic avascular region. And in order for blood vessels to grow, endothelial cells proliferate and also undergo cell shape changes. For example, they elongate. And in order to form an interconnected vascular network, endothelial cells need to branch and also anastomose or fuse. And also concomitant to that, endothelial cells need to form lumen through which blood can flow through. And then, of course, the blood vessels need to be stabilized, and endothelial cells then establish cell-cell junctions, and also endothelial cells deposit basement membrane, and also perivascular cells surround endothelial cells. My lab uses the zebrafish embryo to study endothelial cell behavior during angiogenesis. And this is a movie illustrating the process of sprouting angiogenesis, where it is required to form intersegmental vessels that are formed at the zebrafish trunk. This is a movie where endothelial cells are labeled with membrane M-cherry, and you can see how dynamic endothelial cell membrane is during migration, where it forms many filopodia. And endothelial cells also use filopodia to initiate fusion or anastomosis here, as you can see. And also you can see how dynamic the apical membrane is. So the apical membrane is the inner membrane here. And this is, you can see the dynamics of lumen formation through the blood vessel in angiogenic blood vessels. And when we look at the blood vessels, you can see that no one endothelial cell looks alike. This is a tip cell, and you can see that it is T-shaped. And the following stalk cell is more elongated with protrusions. And the cell at the bottom is more compact, more in shape. So no one cell looks the same. And when we then follow a cell shape, you can see that endothelial cell undergoes extensive cell shape changes. And this is an example where in green is one cell, and it's undergoing lumen formation. And you can see that it then establishes an apical membrane here, and also here, which invaginates into the cell cytoplasm. So as an analogy, it's like the double-walled Bowden cup, where the inner wall is the apical membrane and the outer wall is the basal membrane. And within the apical domain, it's the lumen that carries blood plasma. So I hope from the movies that you can appreciate how dynamic endothelial cell shape is, and this leads to the question of how cell shape is regulated. And a few years back, I decided to investigate the role of actin cytoskeleton in regulating endothelial cell shape changes in angiogenesis. I started by simply generating a zebrafish line where live-act EGFP is expressed specifically under an endothelial promoter called FLE1-EP. And by performing time-lapse imaging, you can observe that endothelial cells generate diverse actin cytoskeletal structures at distinct phases of vessel morphogenesis. During migration and anastomosis, you can see actin and filopodia. And during lumen formation through the vessel, you can see discrete actin polymerization, discrete and transient actin polymerization in the apical membrane as highlighted by the blue arrows. And because several endothelial cells make blood vessels, you can also see actin cables formed at cell-cell junctions. And the cables are maintained at the cell-cell junctions. And in today's talk, I would like to highlight the function of the actin cytoskeleton that is formed at the apical membrane and also actin cables that's formed at cell-cell junctions in blood vessel lumen formation. There are several ways in which lumen can form in angiogenic blood vessels, and in zebrafish, two mechanisms are frequently observed, and I will talk about them today. The first is blood pressure-driven apical membrane invagination, and this is illustrated here in this cartoon. In black, the black line depicts the basal membrane, and the magenta line depicts the apical membrane, and this is the lumen that's filled with magenta. So this process is dependent on blood flow, and which then pushes the apical membrane so that it invaginates into the cell. And when you look at the cross-section, you can see that this is the lumen. This is a time-lapse movie of apical membrane expansion into the vessel. You can see that it's very dynamic. It forms protrusions that we call inverse flaps, because they protrude into the cell cytoplasm. And when you look at the dynamics of the apical membrane flaps more carefully, you can see that there are two classes. There are flaps that retract, and it's shown here in this chymograph where you can see the expansion of the flap, which then retracts. So here again, expansion and retraction. And then we also observe a second class of flaps that expand and then persist, and expand and persist. And these are usually found at the lumen, at the front of the expanding lumen. And we think that that serves to help lumen expansion. And, membrane blabbing is dependent on blood flow. This is under normal imaging conditions where we treated the embryos with one-time tricaine. But you can see the membrane blabbing in the apical membrane. But when we added a higher dose of tricaine, so to inhibit heart contraction and therefore decrease blood flow, you can see that membrane blabbing decreases, shown in this chymograph, and this then leads to inhibits lumen expansion in the vessel. We then examined actin dynamics more carefully in the apical membrane, which is highlighted in magenta here, and green depicts actin, live act. If you follow this region of the apical membrane here, you'll see that it undergoes expansion. And this region is generally devoid of actin. But of course, you also see that there are discrete regions in the apical membrane where you see actin polymerization. And when we take faster movies, you can see that the appearance of the actin around the flaps coincides with the retraction. And you can see it more clearly in the chymograph here, where in magenta it's membrane and live act is in green. During the expansion phase, there's no actin, but actin polymerization begins from at the start of the retraction until the end. Similarly, we also found that myosin II accumulates at the apical membrane. And here, myosin II is in green. And if you look in this region here, you can see that there's enrichment of myosin II in membrane flaps. And when we compared the localization of myosin 2 in green and actin in magenta, we found that myosin 2 recruitment to the bleb surface occurs after the onset of actin polymerization. These co-localization studies suggest that ectomyosin contractility is required for bleb retraction. And to test this hypothesis, we expressed dominant negative myosin 2 specifically in endothelial cells during the phase of lumen formation. And this is an example of a cell in green overexpressing dominant negative myosin 2. And I hope you can see that the apical membrane is blebbing excessively and in an uncoordinated manner so that you do not see a lumen formed. And this is another example. In green is a cell overexpressing dominant negative myosin 2. And here, I hope that you can see that there's enlarged membrane blebs because the blebs cannot retract. And this leads to a dilated lumen phenotype. In summary, blood pressure-driven apical membrane invagination requires a process of inward membrane blebbing that is dependent on blood flow. And endothelial cells respond to these membrane deformation by generating local ectomyosin contractility, so to normalize the apical membrane and to control excessive membrane blebbing and thereby allowing directed lumen expansion. And so that was the first mechanism of lumen formation. I will then now like to go on to the second mechanism of lumen formation, and this is called cord hollowing. Here, endothelial cells are initially organized one after the other, and in this organization, it is known as unicellular vessel. And in this organization, junctions are circular in shape and are separated. But as vessels develop, the junctions elongate, and as it does so, it brings together a convergence of cells. And the junctions then form a linear structure along the length of the vessel. And in this process of cord hollowing, apical membrane domains are formed de novo at cell-cell junctions. And when junctions elongate and converge together, this brings the fusion of separate apical membrane compartments, which then expand to form a lumen. So during this process of junction elongation and rearrangement, we see an increase in actin cable assembly at the cell-cell junctions. We then asked how actin polymerization at the junctions is regulated. We found that the actin nucleation and elongation factor, FMNL3 or formin-like 3, has a role in this process. So these are still images of a movie that shows the dynamics of FMNL3 in magenta and actin in green. At the beginning, FMNL3 is quite cytoplasmic in localization, but as the vessels develop, FMNL3 becomes localized to the cell-cell junctions where actin cables form, suggesting that FMNL3 may be the actin nucleation factor responsible for generating actin cables there. And we then tested this by performing FRAP analysis of EGFP at junctions. So these are already vessels that have formed and they're luminized, and this is a dorsal aorta in a zebrafish embryo, 3-day-old embryo. And when we FRAP the EGFP actin, you can see that there's a high turnover of EGFP actin. And when we inhibited the embryos with a drug called SMI-FH2 to inhibit formin activity, there's an increase in the half-life of EGFP actin at the junctions when compared to the control, suggesting that actin polymerization here is slower after formin inhibition. And we also analyzed the actin cable morphology at the junctions. In control, you can see the actin cables are linear in morphology. But after formin inhibition using SMI-FH2, the cables become more serrated in appearance. So this is the quantification, where in red you can see that actin cables in embryos that are treated with SMI-FH2, they are more serrated when compared to control. So this indicates that actin bundles at the junctions become destabilized after formin inhibition. Then we wanted to know what happens during development when blood vessels are starting to form and develop. So this is an intersegmental vessel from an embryo with decreased formin activity. And we frequently fail to see actin cable formed at cell-cell junctions compared to control embryos, where here you can see clear, distinct actin cables formed as highlighted by the arrowheads. And when we imaged embryos with reduced FMNL3 expression, we found that the junctions are unstable. And this is an example where here is an intersegmental vessel, and this is the DLAV. So it previously formed a junction here. But okay, if you look at the still images here, the junction is unstable. You can see that the endothelial cell starts to make philopodia and migrate over the neighboring endothelial cell. And the lumen that was previously formed becomes disconnected. So, and you can see here in the final still that there's no longer connection between these two endothelial cells, indicating that junctional actin maintains endothelial cells' junction stability. And we confirmed this by performing cell culture studies where we inhibited endothelial cells with the drug SMI-FH2. And in black, it shows B-cadherin at adherence junctions. And I hope you can see that upon inhibition of formin activity, there's destabilization of B-cadherin clusters, leading to the detachment of endothelial cells from each other. And this is in clear contrast to the control, where you can see that V-cadherin clusters remain at cell-cell junctions and cells remain attached to each other. We then examined the organization of the junctions in zebrafish morphins, where there's reduced FMNL3 expression and activity. So this is in a control situation, embryos that have been stained for V-cadherin and tight junction molecule Z01, and this shows actin. And this is just an outline of the junctions, and you can see that the vessels show a multicellular organization of junctions, and the junctions are linear and are connected. But in FMNL3 morphins, the junctions appear more circular, and also the junctions are disconnected from each other, indicating that cell junction elongation failed to occur. As a consequence, because of reduced junctional actin formation there, the vessels of embryos with reduced FMNL3, or in embryos with endothelial overexpression of dominant negative FMNL3, and in embryos that have been treated with SMI-FH2, many vessels fail to form lumens, while control of embryos show clear lumen formation, as shown here. Just to summarize, in the second mechanism of lumen formation called cord hollowing, actin formation at junctions strengthened V-cadherin adhesion, and it strengthens and stabilizes V-cadherin clusters, thereby allowing junction elongation. And because apical membranes need to converge together to fuse and expand, when junction elongation fails to occur, the lumen fails to form. And with that, I would like to thank my postdoc supervisor, Holger Gerard, and also a previous PhD student in his lab, Veronica Bala, whom I collaborated with to work on the lumen expansion project. And as I said earlier, I moved to Weekend Center for Developmental Biology about a year ago, and I would like to thank my lab members for helping me set up my lab and start the projects. Thank you for your attention.